My name is Beth Simony. I'm a senior group leader at the imaging platform, the Bird Institute at Cambridge, Massachusetts. In this video series, I'm talking about morphological profiling. In part one, I introduce you to what that is and how we go about generating data sets for morphological profiling. Here in part two, I'm going to take a data set that was introduced in part one, where we have drugs from different mechanism of action classes, and then show how, when we get morphological data sets in the lab, we actually practically go about looking at them and trying to do biological discovery. A brief reminder for those of you who may not have just watched part one, morphological profiling is essentially the idea that whatever objects we have with whatever treatments and whatever kinds of stains, if we just extract as many features as we can possibly think of, we can then cluster the cells or wells that come out of that to try and learn connections between different groups. And so we might learn that there's a connection between two different drugs, or between a couple different genetic perturbations, or between certain patients. And so the secret to this technique is just creating lots of measurements. A few things that are critical, though, for this technique to work is, of course, though, that your objects are found and measured correctly in the first place, and we have some guidelines in the video description as to how to do that. And I also just want to point out that these sorts of connections are amazing to discover, but should then be more deeply explored. Any two things that seem correlated or anti-correlated or uncorrelated, when you dig more deeply into them, there could actually be lots of stories hiding there in the data. And so to use these sorts of analyses to create hypotheses for you to dig into further. And the final caveat is just that morphological profiling is a huge, rich field that is constantly evolving and has methods all of its own. So I'm only going to show you today a tiny slice of what is possible with these sorts of techniques, but this is the tiny slice that in practice we typically get started with. In practice, how we actually go about doing this is by finding and measuring things in Cell Profiler, although certainly you don't need to use Cell Profiler for this. Anything that generates measurements will do. In our particular workflow, what we do is for every site from every well of every plate, we create its own set of spreadsheets and use a tool called Cytominer Database, which is a command line Python tool, to then re-aggregate all of those CSVs back together so we can have information about every cell all in one container. We then use a tool called PyCytominer to create per well means. And I've talked a little bit more in video one about why we use those. If you're not that familiar with the command line and would like to avoid it, you can get a reasonable first pass at data to do morphological profiling on by having Cell Profiler turn on per image means and getting a per image mean spreadsheet, and then using Morpheus's collapse function to turn per image means into per well means. But I will warn you that you're not going to get exactly the same results that way, because if you have one image with 5 cells and one image with 500, Morpheus is going to average their means exactly as if they were the same. Using the command line version, you're going to get a weighted average that is probably a more accurate representation of what's going on in your experiment. So if you're willing to do just a tiny bit of command line work, you'll actually get the highest quality data. Likewise, once we have our per well means, our typical workflow is to use either Cytominer, which is written in R, or PyCytominer, to then add our metadata, normalize, and feature select. And again, you can go back to part one for information about why we do these particular steps. This Jupyter notebook will be linked to in the video description, and it shows how we go from per well mean to a final feature selected file to use. And it's zoomed out here because I don't really want you to focus on the details. I want you to just note that there's actually only 2 required steps here, and at most 4 steps to go between that. So even if you're not very comfortable with using these sorts of command line tools, it's actually not that difficult, and there's not that much code that needs to be written. The first step is just to import your libraries and your data. In step 2, if you didn't already put your metadata into Cell Profiler or whatever you did to make the measurements, you need to then put that metadata onto your per well means. So in this case, we had 2 different levels of metadata, and so we had to do 2 levels of annotation, but typically you would be doing 1 or even 0 and might be able to skip this step altogether. Likewise, these steps are just data cleaning and are not necessary unless you want to make a CSV that you can use to explore your per well information in something like Excel or some other spreadsheet program. If you're just then going to Morpheus, you don't need to do this kind of data cleaning at all. And lastly, the actual normalization and feature selection, which is only one line of code for each and then a line to just export out the intermediate results. And so even if you're not very comfortable using Python in a command line or in a notebook, the libraries that we've written can actually make it pretty easy for you to generate the highest quality data in order to actually do morphological profiling. So let's now take a look at how we actually, in practice, do discovery in Morpheus from one of these feature selected files. So this is the Morpheus interface. I have a couple different file type choices. If I have a CSV that I got from Cell Profile or from Excel or from somewhere, that can go straight into Morpheus. Although if you're going to do that, you want to arrange your data and possibly move your columns around in something like Excel so that things like your file paths and your metadata are all at the very beginning, because what I'm going to do here is in a CSV is I'm told that I have to start the data matrix and everything to the left is going to be one kind of annotation and everything above is going to be a different kind of annotation. And so if I have metadata columns in here, it's very difficult to then sort of get them back out. And so you want to rearrange your columns. It definitely works to import them though, although compared to everything I'm about to show you where we start from a GCT file, if you want to do things the same way here, you'll need to go to this Tools menu and hit Transpose so that you have columns that are wells and rows that are features. Typically though, we just start from GCT files instead, which are made in that same Jupyter notebook that I demonstrated. And so when I load data in this way, it's already in the form that I want, which is that features or measurements are rows, and that you can't tell this yet, but you'll have to take my word for it, that wells are columns. So the first thing is, let's make it so that you don't have to take my word for it. If I go to this options gear, I can change the annotations on my columns instead of having this ID, which isn't particularly helpful. I can add things like the compound, the dose of the compound, the MOA, the plate, the well, whatever I happen to already have. I can control click or right click on any of these and give them a color, which is nice for sorting. For something like a concentration where it's a number, it will scale linearly. Otherwise, it will just give each value a different color, as you can see here with plates. So as I scroll through here, you can see the MOA and compound colors change, as do my plates. Now, if you didn't just come from watching part 1, let me give you a high-level overview of what we want. What we want to look at is something where we can create an overview of how similar each well is to each other well within our entire experiment. And so what we're going to do is create what's called a Pearson correlation similarity matrix. And let me quickly graph what a couple of these wells look like in feature space in order to actually, I think, give a more intuitive understanding of what that means. So I'm going to shift click to select all of my features. I'm going to sort this by plate and grab wells B2 and C2, which are DMSO treated, as well as C5, which is drug treated. Now if I come into Morpheus here, into my tools menu, I can make a chart of these. I can make the chart fit, and I can label it with my well. So comparing B2 to C2, which are both DMSO treated, or B2 or C2 to C5, which is drug treated and these are DMSO treated, what at least jumps out to me is that we have a pretty tight correlation here and not a very tight correlation in either of these charts. So if I did a Pearson correlation, this would probably be something along the lines of 0.9, and these would probably be something around 0. And so the similarity matrix we're going to make is just going to be this, sort of taken one step out. We're going to, for each pair of columns, calculate the Pearson correlation between these two columns across all of their features, essentially the Pearson correlation of this line, and then plot that. So in practice, what this looks like is this. So here is our B2 and C2, and if I hover over the square that's at the intersection of those two, I've got B2 on the rows and C2 on the columns, and this little bar up here has nothing on it now, but when I go back down, it will show you exactly where I am at all times as well as the value. We're seeing a correlation between those two wells of 0.9. Whereas B2 to C5, which I said didn't seem very correlated, we have a Pearson value here of 0.2. If I do C2 to C5, it's a little bit better, it's 0.29, but it's about the same. And so essentially what we're looking at is just lots of Pearson correlations, now with that becoming the value that's graphed. So now that we have our similarity matrix, what do we want to discover? Well, in this experiment, we have a number of different drug mechanisms of action, or ways we think drugs work. And within each mechanism of action, we have a number of drugs that we think have it. And so the major question in the original experiment was, can we see that drugs with similar mechanism of action look similar by morphological profiling? And there are a couple different ways to do that, but I think the easiest is to sort. Before we do that, though, we want to come up to the gear and to the display, and then say that we want to link the rows and columns. And that means as we change our sorting in one axis, it will actually change in both axes. And so it will make it easier to look along the diagonal for what we want. So in order to say, are our drugs sorting by similar by mechanism of action, I'm going to sort by first mechanism of action. And then by holding down Shift, I can introduce subsorts and say that I want to look by compound. And just in case it matters, it may not, by concentration. And so now I have my data sorted in that way, and I can start looking for patterns. So let's look at our first mechanism of action here, actin disruptors. We've got 3 drugs in it, cytochlasin B, cytochlasin D, and latrunculin B. And they're the same along either axis. So as always, when I go to the absolute diagonal, it's going to be 1 because we're comparing each well to itself. But what I can see by eye and by mathematical value is that particularly within a dose point, our actin disruptors are clustering within each cell very well, at least by drug. So cytochlasin B all looks very much like cytochlasin B, even across a couple of different doses. Cytochlasin D looks like cytochlasin D, and latrunculin B, again, across a couple different doses, but most tightly within dose, looks pretty similar. In terms of an overall mechanism of action-based similarity, there's a reasonable similarity. Certainly if we start looking off diagonal between cytochlasin B in the rows and latrunculin B in the columns, then we can say it's still pretty reasonably similar, not that much less dissimilar than each drug is to itself, but cytochlasin D seems to be not quite as related to the other 2 drugs. These values are not quite as high. And we can then just sort of look by other mechanisms of action to see if it seems that pattern typically holds. So our aurora kinase inhibitors all look very similar to aurora kinase inhibitors. Likewise, our cholesterol-lowering drugs, of which there are 2. And if we look even further off the diagonal, not just off diagonal between individual drugs within a class, but now across classes, we can start making comparisons and making conclusions about if different classes of drugs look similar to each other or not. I'm going to skip through the DMSO for a minute here. But we now have DNA damagers and DNA replication inhibitors. And those all look pretty similar outside of, it seems like maybe one well that had an artifact, and that's why we always do multiple wells. And again, the text is probably pretty small to read on the screen, but as I scroll through and see the different mechanisms of action, what you can hopefully appreciate is that we have large blocks of red. And so what that says to me And what that said to the scientists who originally did this experiment is that yes, using these sorts of measurements, we can absolutely group drugs into their mechanism of action classes with pretty high confidence. Now, I want to return to this DMSO because, again, it's seeing a wide variety of phenotypes, some of which are correlated pretty tightly, but not necessarily to each other. And we have a whole range that's here in the middle. So depending on how our data was normalized, we would either hope that all of the DMSO would be tightly correlated to itself if we had normalized across, say, a whole plate's worth of data, and so DMSO was a phenotype. But if we had normalized to DMSO as our baseline, which is what I believe was done in this experiment, we would hope that DMSO essentially just looked like noise, looked like nothing. So what are we actually seeing here? Well, if I zoom in a little bit, what you might be able to start appreciating is whenever I have these big blocks, my plate things are pretty similar. So let's sort by MOA and then plate. And yes, where I'm seeing big bricks of red in the DMSO, it seems to pretty tightly be correlated to plate. And as I hover over the batch names, or the plate names actually have batch names in them. And it looks pretty clear to me that, for example, at least sometimes within a batch, which was all taken in this case during a week, so this plate all were processed together, these are pretty tightly correlated. And we can see here, these are the DMSOs of week 8, and so on and so forth. One unfortunate artifact, or one unfortunate difficulty of cell painting being, and these sorts of morphological profiling assays being very sensitive, is they are very sensitive to a lot of things, even sometimes things you wouldn't want them to be sensitive to, like which week was your plate run, in which batch with which sort of aliquot of reagents. One other way that we often like to look at this data is not by sorting it, but also by hierarchically clustering it and seeing if anything interesting seems to fall out. And so in this case, we have a pre-computed similarity matrix, so that's what I'm going to tell it to use. And I'm going to tell it to cluster it across rows and columns. And so when we look at the hierarchical clustering data, one thing that we see is just this whole left half of it is blue, which means is our DMSO. So even though we do see these sort of big bricks of DMSO, just lots of red, our DMSO is still mostly more similar to itself than it is other drugs, although that certainly doesn't hold everywhere. For example, there's some DMSO treated drugs here, or plates here. In terms of then linking mechanisms of action, you can see that hopefully that these blocks are pretty monocolored. We typically have a strong block of color as opposed to the colors being sort of all mixed together the way they currently are for plates and wells. Now that's really exciting news because that says that more so than plate or well position, what's really driving how similar wells are to one another is their drug mechanism of action class. So that's where adding color here can be really helpful in terms of making a biological conclusion. And so we're seeing a block here where things like protein synthesis, I don't know what epithelial is, and cholesterol lowering drugs, as well as actin disruptors, are all kind of similar to each other. I'm not sure why those would exactly be, but that's for the next step of biological discovery. And here we have microtubule stabilizers and destabilizers actually looking relatively similar to one another, which sounds slightly counterintuitive up front, but when you realize that microtubules dynamically assemble and that disrupting either of them is just going to overall wreak havoc on the cell, it's maybe not so surprising. And so with a hierarchical clustering, again, we're able to make more biological discovery. I want to return back to my features here just to do another couple things. So one experiment that was present in the original paper and that I mentioned in part 1 of this video is the idea that actually a lot of this mechanism of action can be discovered even just from looking at nuclear features alone. So if I want, I can filter to looking at just nuclear features and then run my similarity matrix again. So I can say, only give me things that have nucleus in the name. And I can then run a new similarity matrix. Again, I'm going to link my rows and columns. Zoom out. I'm going to do the same sort I did before, MOA, then compound, then concentration. And yes, pretty similar to what was found in that paper, looking with nuclear features alone actually creates a really strong ability to predict mechanism of action. So even, again, things down here in the categories like protein synthesis, we're pretty strongly able to tell that those drugs are all tightly correlated to one another. Get all of my features back here now. One other thing we often can do when we want to sort of look at an unsupervised clustering type thing is perform a t-SNE. And exactly how one best creates one of these t-SNEs is way outside the scope of this. So there are other resources you can look at. We'll link to some in the video description. What t-SNE is essentially trying to do is to create a different way of comparing the similarity between individual columns so that we can look at that in a different way. Maybe Pearson doesn't give us the best results, so t-SNE is going to do that differently. And so I can create a chart here where my T1 and T2 are done and say that I'd like to look at a t-SNE embedding. And so look at T1 versus T2. And my t-SNE has grouped, instead of having a hierarchical clustering, what we have here is a grouping instead. I'm going to make the symbol size a little bit bigger so this is a little bit easier to see. So right now we're just seeing that there are all of these different groups, but we don't really know what's going on in each group. But I can change my color to say be by mechanism of action. And what I mostly see are relatively tight clusters. Certainly not perfect. This blue guy is all the way off over there, but we have a pretty tight cluster of red, of purple, of blue. And if we look at drug instead, we do see that multiple drugs are living inside the same mechanism of action cluster. But if we change our sorting to look for plates, the plates are very well scattered, indicating that, again, mechanism of action is stronger than things like plate effect in this experiment. A couple other kinds of things we often want to do in Morpheus. One of them is just for quality control. And that is to change the actual visible values here to make sure that we don't, for example, have any features that are really high or really low and otherwise driving what our Pearson correlations and t-SNEs would be. There's not a ton of evidence for it in this data set. This is a pretty clean data set. But by saying, instead of having a relative color scheme, that it should scale all of these, say, -1,000 to 1,000, or -100 to 100, what I can do is I can highlight for myself if there are any columns or rows that are problematic. Now, a column that's problematic might be a well that's just sort of full of debris or crud, and a row that's problematic might just be a feature that behaves badly. Now, in general, we always want to err on the side of leaving data in, but if we have, say, a whole lot of features that are in the sort of -5 to 5 range, and these features are in the millions range, that's going to throw off our Pearson correlation so badly that our data becomes useless. And so this sort of quality control is never a bad idea. Put my relative color scheme back. And the last thing I want to do is just demonstrate a marker selection. The most common question that we're asked after we help somebody with morphological profiling experiment is, cool, you're able to see the difference between my group A and my group B. What features are driving that? Now, unfortunately, it's not always as clear as you would hope to say, oh yes, well, it is this feature and therefore this is the biology that's causing the difference between, say, your drug A and your drug B. Often these are a little bit hard to interpret, but it's good to know how to do it so that you can at least look to see if there's something obvious. So by going back to my tools and going to a marker selection, I like to do a t-test. And so I can do t-tests between any sets of things. So maybe what I'll do is I will t-test based on mechanism of action, my controls against, say, DNA damage. Now what this will do is it will tell me between all of my controls and my DNA damage which features are most different. And so those might be ones that I'm interested in thinking about whether or not they contribute to the biology. Now here we're sorted by the t-test score, which is going to have a high absolute value for either thing, but what we really want to sort by is the p-value or false discovery so that either very high or very low correlations show up. And what's surprising to me is that for a DNA damage, the top several features that are most different are actually cytoplasmic features, cytoplasmic texture features in the tubulin and actin channels. That's definitely not what I would have predicted a priori. Now, eventually, we start getting some nuclei features, but we really do have quite a mixture, and that's why sometimes doing these marker selections can be a little bit unsatisfying, because even in a case where we have a strong sense of what we think the features ought to be, it's not always necessarily what actually is driving this. And so you need to be a little bit careful with doing these, but sometimes you get an obvious answer such that, the top 20 things are all nuclear features or are all features that are related to a particular channel. And in that case, you have a really strong place to start from in terms of guessing what actually is happening in your biology and therefore what your next experiment might be or what are good things to do to validate it. In conclusion, morphological profiling can be a really powerful way to explore cellular phenotype data and find connections between kinds of samples. Profiling is an incredibly complex and evolving field. I've shown you just one of the kinds of things you can use these techniques for, but point-and-click tools can help you create a first-pass head check. We've linked here in the video description more resources of where you can get started, but you can also work with your friendly local bioinformatician to then follow up these sorts of first-pass analyses with deeper work in order to really nail down your biological discovery and use all of that mathematical information found in your microscopy images to its fullest extent. And with that, I'd like to thank the members of the imaging platform, both in my lab and the Carpenter Singh lab, for creating these tools and for pioneering these workflows that we now get to share with you. This video series is sponsored by the Center for Open Bioimage Analysis, and we also want to thank our other funders and our biology collaborators. Thanks, and good luck.